Hi, everyone, and welcome to episode 32 of the Cancer Researcher podcast, brought to you by the European Association for Cancer Research. I am your host, Dr. Alexandra Boydor, Scientific Manager at the EACR, and in this episode, we will discuss some research featured in the EACR's Highlights in Cancer Research. To set the stage for today's conversation, I start by saying that many cancers are innervated, and in recent years, the neuronal component of the cancer microenvironment has received increasing attention. At the same time, the metabolic plasticity of cancer cells is becoming increasingly recognized as essential for cancer progression. Dr. Simon Grelet from Mitchell Cancer Institute, University of South Alabama, and Dr. Gustavo Ayala, physician scientist at University of Texas, are two of the scientists that lead research at the border of these two research fields: cancer neuroscience and cancer metabolism. More exactly, they are looking into metabolic reprogramming as a consequence of tumor innervation. Previous studies from their groups demonstrated how cancer innervation and cancer cell plasticity are intimately linked. More recent research from their lab is looking into the cell-cell transfer of mitochondria, which has recently become a subject of investigation as it has crucial functions in health and disease. In this episode, we will discuss their paper entitled Nerve to Cancer Transfer of Mitochondria During Cancer Metastasis, published in Nature last year and featured in the EACR Highlights in Cancer Research. Let's dive into today's topic. Hello, Simon and Gustavo, and thank you so much for allowing us to glimpse into your research.
Hi, it's a pleasure being here.
Thank you for the invite. I'm glad to be here too.
One of your recent papers is looking at the impact that tumor innervation has on the metabolic plasticity of cancer cells and subsequently on the aggressiveness of breast cancer. What made you direct your attention to the intersection of cancer neuroscience and cancer metabolism?
I think that's a long story, and Gustavo may want to start because he was a pioneer on this field.
Sure. I mean, it's 27 years ago, we did our first paper, and that paper just told us that cancer nerves played with each other, talked with each other, were good for each other. They made each other grow more, and then we found out that you cut the nerves, tumors don't form. And we started to find a trans-species signature of nerve effect, and that was alterations in metabolism. Essentially, if nerves are not there, cancer follows the Warburg effect. But if nerves are there, they become metabolically efficient. You cut nerves, they go back to being glycolytic. And so for the longest time, we looked at how this happened, and we really couldn't find a transcriptomic profile that would justify that. And then I met Simon. Take it from there.
So basically, I would say in my end, I started to study cancer innervation way after Gustavo, actually, but it was because of Gustavo, because years ago I was studying cancer plasticity. So most of my work revolved around the cancer progression and especially focusing on cancer metastasis because that's related to cancer-associated mortality. And a few years ago, I was studying cancer plasticity and figured, looking at some transcriptome data sets, that cancer cells actually express neuron-related molecules. They express molecules that are supposed to be expressed in neurons, typically that you can find in the brain, axon guidance molecule. And I was studying cancer cells on a petri dish, not even on a mouse. And that's where I started to scratch my head. And that's where I stepped in the field of cancer innervation and looking at what have been done in the field. Actually, when I started, there was not many papers, but there was some of these seminal papers from Gustavo about axon guidance molecules that are secreted by prostate cancer cells and that attract neurons into the tumor bed. And that's where I really started to think about various neurons in cancer. I never thought about that before. I was thinking about, obviously, the immune cells, the fibroblasts, but I never thought about neurons. And still, when I talk about my work right now, I meet people that have the same reaction that I got a few years ago about neurons in cancer. But now it's more and more accepted. So that's how I stepped in the field. And when I established my research laboratory, I entirely focused in this topic because I think that's a very important and uncharted territories in cancer research.
As you highlighted, Simon, this is a new field of research. I mean, cancer neuroscience is a new field of research, but it's also a booming field of research. So what would you say is the element of novelty that your study brings?
The novelty lies on what we know actually, because what we know for years and from what pathologists will say about cancer innervation is nerve density in cancer is associated to poor outcome in cancer. So that was known. That is a fact, but what we brought was the novelty of our work relates to the mechanism that are quite surprising in their nature, because we were all looking to signaling pathways that could be activated at the nerve cancer interface. And what we found was very, very different. So I think that the novelty which is brought by the paper, if Gustavo have something to
Yeah,
say about that.
I agree totally. It's novelty and impact. So for the past 50 years, we have been focusing on genes. Cancer is defined as a genetic disease. Changes in genes produce cancer, right? And make cancer more aggressive. And I think that's true, but it's not the whole story. And what we see here is the closing of the loop in this history of cancer nerves. Cancer doesn't turn on genes or turn off genes. The modulation of the energetic metabolism, and the impact of which is it makes cells metastatic, it's humongous, is through organelle transfer, horizontal mitochondrial transfer. And it gives you a whole different view of what cancer is and what we're going to need to do to be able to modify cancer's behavior. Maybe targeted therapies will have their role, but also molecules that modulate nerve function. It's really important to understand that the modulation of nerves will be necessary to let other therapies become more important. So again, if you think about impact, you stop chemotherapy, you stop radiation therapy when you start affecting nerves because you can't take that back. But you're leaving the most important element that cancer uses to kill. So all these therapies are going to become much more efficient if we learn how to modulate nerve function and understand that that modulation needs to happen through non-transcriptomic ways.
As you say, Gustavo, that's bringing a new layer of understanding of what cancer is, because we knew, for instance, that non-cancerous and cancer cells can communicate with each other, can activate signaling pathway. We knew that. And then we found they can even transfer some molecules. For instance, we can transfer some metabolites. We knew that it's kind of fueling the cancer by giving some metabolites. You give a battery to the cancer cells, but here on this particular setup, it's like we transfer the power plant. We transfer the way of generating energy from neurons to cancer cells, which is much beyond what we thought. And taking in consideration that neurons have extremely good energy producing organelles, instead of just sending the fuel, we send the factory and the plan of the factory as well in terms of metabolism.
And it's not just the how it happens, it's the impact it has. This horizontal transfer really defines cancer aggressiveness. This is what makes cells have metastatic potential through all the mechanisms that we described in the paper.
I'll admit that this is new territory for me. Although I find it fascinating, I can't say that I understand the nitty-gritty details of nerve cancer cell interplay. So I was wondering if you could please explain the mechanisms associated with cancer nerve metabolic dependencies in cancer progression. And I know this is like a huge question, but from what I understand from your paper, cancer sort of boosts the neuronal mitochondria, and then in turn, neurons transfer those mitochondria to cancer cells.
That's right. So that's a bidirectional discussion. There is really like this connection between cancer and neurons, which is occurring. The power of the data we generated also rely on multiple data sets that Gustavo have generated in his lab, I have generated in my lab, in rat, in mouse, in humans. We always came to the discussion in every data set that if you block the nerves, you block the metabolism. So that's the power of the finding and the nerves on this crosstalk. So that's one aspect. And another aspect is during this process, we were able to capture like the big pictures of what the nerve does to the cancer by denervation, especially by Botox denervation that does block every nerve. So that's a way we need to study the impact of the nervous system into the cancer biology. And it was always pointing, whatever the model, always pointing to the metabolism and always pointing to the mitochondrial metabolism, mostly bringing to the down direction. Very striking, every data set. And what was needed to confirm the direct relation between the nerves and the cancer cells was also to do like more simplistic models of nerve cancer crosstalk that we can study in more detail and to see if it's a direct mechanism or if the neurons are just a bystander of something else, given the complexity of the tumor microenvironment. And that's where we developed a model of neuronal stem cells to cancer interaction, where we mix neuronal stem cells and cancer cells. And that allowed us to scale up the co-cultures to study in more details the crosstalk. So that's what we call the SVZ models in the paper. And it did allow us to get this unprecedented view and generating high quality and high level nerve cancer co-cultures to solve this problem.
And did you get the chance to look at what regulates the directionality of this transfer? Like, do neurons actively donate mitochondria, or are cancer cells effectively extracting them via induced mechanisms such as tunneling nanotubes or synapse-like structures?
It's both.
Okay.
I have a picture where you can actually see a neuron, right, donating mitochondria to a cancer cell. And at the same time, on the other side of the image, you see cancer cells tunneling nanotubes and stealing mitochondria from the cancer cells. So it's bidirectional. And again, if we go back in time, it's really funny, but this is exactly what the first paper said. I love this picture because it takes me back to that first paper that just said cancer cells and nerves like to play with each other. And so what we know today is how they play. We've known for a long time that they were good for each other, that there was a symbiotic relationship. And by the way, Simon did some experiments where we defined that the supernate and secreted factors were not as important. So we started concentrating on direct transfer. And I also have another picture that's not in the paper where you see membrane to membrane transfer of mitochondria. You see membrane dissolution and the mitochondria going. So we think that it's more important for mitochondria, which are relatively big, although you can have huge, and I have anecdotal data from other investigators that mitochondria can be transferred through large exosomes and different types of molecules, right? So I don't believe that there's anything in nature that is exclusive. I think that there are preferential mechanisms. And I think that the preferential mechanism of transfer requires direct contact.
Yes, especially when we talk about neurons, because as you say, we tested like distant versus direct contact. And in terms of nerve cancer crosstalk, it is contact mediated mostly. Even if we see some non-contact mediated that's significant, it's not like the majority. That's like flying fish.
You need to understand the nervous system is the perfect system to do direct contact transfer. The nervous system, and we really don't think about it this way, it touches every single cell of the body. So we think that nerves go in end and there's a space, and no, nerves touch or are in sufficient vicinity of all the cells in the body. The other thing is a lot of effort has been put into what do neurotransmitters do for cancer? Because we're thinking of nerves as nerves. We have defined nerves by the fact that they have synapses and transmit neurotransmitters, right? Through synaptic junctions. That's our definition of nerves. But we tend to forget that nerves are also endocrine. Neuropeptides can't go through synapses. They have to be secreted into the microenvironment. And so what we're finding now is a new function for the nervous system. And I'm going to go on a limb here. But we think, and we're going to try to prove, that this is physiology, that this is not a cancer-exclusive process. We think this is how the nervous system regulates the energetics of the normal body. But we have ways to go.
Which would be in line with cancer cells being able to hijack mechanisms, which has been seen in other areas of cancer research before.
Yes, that's a unique opportunity where we discover the physiology through the lens of the disease, which means we have the tip of the iceberg and there is all the other part to discover. And that's what is exciting about this project. As you say, cancer cells, they never reinvent anything. They just hijack. For a long time, I've been studying epithelial mesenchymal transition, which is an embryo development, wound-healing development hijacked by cancer during metastasis. We discovered that before the function in metastasis, but here we are doing the reverse direction, and that is super exciting.
Your study has a double value, the development of methodology and also investigates the effects of mitochondrial transfer on cancer cell biology. And you developed the mitotracer to permanently label the recipient cancer cells after mitochondrial transfer from neurons. And you've mentioned during this conversation that this mitochondrial transfer has a huge impact on the aggressiveness of cancer. So I was wondering, how do neuron-derived mitochondria confer increased metastatic abilities?
Okay, so that's like two questions in one. Let's break down what we developed and what mitotracer is and how we use that to understand the impact of the transfer of mitochondria and how did we leverage this technology to better understand the mechanism. So what you need to understand about the mitotracer is when we went to this metabolic crosstalk between cancers and neurons, that was super interesting. We were able to document the transfer of mitochondria. There is several ways to do that. You can simply label genetically mitochondria with a fluorophore like a GFP, and then you can see this mitochondria going from one cell to another cell by microscopy imaging, for instance. But there is some limit on this methodology. The first limit is your signal is not conditional to the transfer. So it's very difficult to curate the data. And actually, most of this green signal will be from your donor cells, and you need to identify some of this green signal going into non-green cells as an evidence of the transfer of mitochondria. So that's difficult to analyze, and it's taking time. And the other aspect, which was the major bottleneck for this study, because we made the hypothesis that the transfer of mitochondria is driving cancer aggressiveness and will be one explanation of why highly innervated cancer are more aggressive, is that when this mitochondria are transferred to the recipient cells, the signal of this mitochondria disappear over time, is diluted because the transfer in mitochondria is not labeled anymore. Because the label that you use to label this mitochondria is on the nucleus on the donor cells. So on the nucleus, you produce the reporter that goes to the mitochondria, it labels them, these mitochondria are transferred to recipient cells, and ultimately they are integrated into the recipient cells, but the signal is gonna fade away. And that was a major block to test our hypothesis about the roles of this transfer in changing the phenotype of recipient cells. Because we were not able to clearly distinguish recipient versus non-recipient cells in our system and isolate the contribution of neurons versus the contribution of neurons transfer of mitochondria as well. So that's how we address this problem by developing this technology that permanently mark the recipient cells. And that's what we call mitotracer. We're going to develop that in details, but basically it consists on a pair of scissors and a light switch. So what do you have? You have your donor cells. They are labeled on their mitochondria with a pair of scissors that can make some genetic editing, that's a pre-recombinase. And when these mitochondria are transferred to recipient cells, they activate a light switch and they do some genetic editing in the recipient cells. This is activating fluorescence, and as a result, your cells are permanently marked. And it's kind of stamping a GPS into your recipient cells. It's like you mark these cells with a GPS, and then based on this fluorescence, you can follow their fate. And that's what we use to understand the roles of this transfer in cancer aggressiveness in general. So ultimately demonstrating their role in metastasis through lineage tracing, because we were able really to follow the fate of these cells and their progeny over time. So that was technology-wise how we enabled that.
I was wondering also if we are to look a bit beyond the increased metastatic potential in the mitochondria recipient cells that you've demonstrated so nicely in the paper, and then the subsequent impact on cell invasion. I was also wondering if you noticed any impact on cell signaling and or the tumor microenvironment. For instance, do transferred mitochondria alter signaling pathways or immune evasion in recipient cancer cells?
There's another paper that is showing that cancer cells actually steal mitochondria from T cells and donate their defective mitochondria. Essentially, the T cells are there, they live, but they're functionless. And that's a big component of the immune inhibition. So what we're seeing is a repeat of non-signaling mechanisms that regulate cancer.
Yes. And to clarify, in our specific biological context, using this tool, we were able to get some surprising way. We discovered like the impact was not what we were expecting actually. And you mentioned signaling pathway. That's not exactly what we found. The first striking observation is in the lab, as soon as we got this recipient versus non-recipient separated into different co-cultures, we made many clones, and we found that the recipient cells were growing on a very different pattern. That was the very first observation. My tech was concerned about the recipient cells not being healthy because they were not attaching to the bottom of the plate. They tend to grow by themselves and detach. And looking more carefully, it's actually these breast cancer cells, they were forming what we call mammospheres, which is a sign of stemness properties. That's what stem cells do. They grow in a surface independent manner. And that's what we found very early. We found these recipient cells behave differently because they like to grow in 3Ds instead of growing in 2Ds, like just by themselves. That was the first observation. I think this data set about recipient cells being more forming metastasis, when we looked at the more detailed mechanism, the first obvious experiment was to test the invasive capacities of these cells. So we took the recipient versus non-recipient on an in vitro setup, on an invasion assay setup, and we did not see striking difference in the invasive capacities per se. And it was like a little bit confusing at that time because these cancer cells, when they receive mitochondria, they form more metastasis, but on the same time, they are not more invasive, or there is not big increase on their invasive capacities. So that's when we decided to take these cells back to the animal. If they are not more invasive in vitro on a simple setup, are they more metastatic when we separate them and re-inject them into the animals? And we found, yes, they do form metastasis. They are not more invasive, but they form more metastasis. It's actually exciting data at this point.
Yeah, I think that was the breakthrough. But also, it's not that we didn't look for signaling mechanisms. And actually, I personally looked for 15 years before we found this. So when you look at transcriptomic data, you see the result, but you don't see the how. And this data is available, so anyone can go and mine it. Maybe I just missed it. So you see, man, 25-year-old data. You see what happens, but you don't see how it happens or what led to it. And I think for me, the importance of this is, well, finally, when we started looking somewhere else, we found that it was non-signaling mechanism.
Just to clarify that metastasis is not only about invasion. And people always think metastasis as a matter of invasion, but actually the metastatic cascade is much more than that. It's a very complex journey for the cancer cells. And cancer cells, not only they need to be invasive to leave the primary tumor, but most importantly, they need to survive throughout the process of metastasis and ultimately outgrow on secondary sites. That could be very different from the primary sites. For instance, a primary tumor in the breast will be on a very different environment than a metastasis growing on the brain, where, for instance, the metabolic environment and the glucose resources are much more limited. And another aspect is when these cancer cells leave, they have to go through the bloodstream, like as a highway to disseminate to the organism. It's a very unnatural environment for a carcinoma cells, which is of epithelial origin, which usually are always growing on a substrate to evolve on a liquid environment. And there is two major bottlenecks in the blood to metastasis. It's the oxidative stress and the shear stress. And when you look at the data, 99.9%, even more, of cells that reach the blood will ultimately not form metastasis. And the reason behind that is they die during the process. They cannot survive this stress. And what we found is the transfer of mitochondria provides the recipient cells with this metabolic plasticity that we know it's associated to resistance to oxidative stress, resistance to shear stress, that are two main factors of metastatic attrition during metastatic progression. And I really want to bring that to your answers because it really changed the way we think about metastasis. Maybe we should not target the ultra-invasive cells that are running very fast, because at the end, 99.9% of them will fail. There is this subset of cells that are not only invasive, but also plastic in terms of their metabolism, which not only run fast, even if it's not like the ultimate requirement, but survive during the journey of metastasis. That's these cells that we need to therapeutically target. And right now, most of the approach is really to target invasion. And we know it does not work very well.
And this is Darwinism at its best. This is natural selection. The cells that survive are the cells that have the ability to survive. And that's what the mitochondria does. And we still have ways to go to understand how it does. I mean, the metabolic plasticity is obvious, right? But how it affects the capacity to survive shear stress and oxidative stress, we still don't know. That's the future, obviously. We've been doing basic science for so many years. I think it's time to concentrate a little bit in the clinic right now. I think that knowing what the mitochondria does to influence oxidative stress, it's good. We will do it. But I think that we know already enough to understand that nerves do provide cancer cells with, if you want to call it, superpowers. Cells become super athletes through this. And I think that neural modulation needs to be a part of therapy as soon as possible. And we are lucky that there are so many neurotoxins in nature and in warfare that can be tested to see how to modulate. And again, with a clinical emphasis. The other way to see this is the nerves are a road. They're super highways through the body that epithelial cells and all other cells gladly accept. They accept the guidance of the nerves through mitochondria and many other mechanisms. So we have to start thinking about the neurotropic nanomolecules where we can put whatever we want to put inside, targeted therapies, chemotherapy, whatever, because this will get to every single cancer cell in ways that we cannot do today. It's actually not the blood vessels that do this delivery. It's actually the nerves. So I want to emphasize that it's good to know the ultimate mechanism, but sometimes we get stuck there. And I think we have a clinically targetable element right now, and we have to start designing, and we're looking for collaborators. So if anybody's interested, email, call.
You do show in your paper that denervation reduces invasive tumor progression in your models. So how feasible do you think it is to therapeutically disrupt nerve tumor mitochondrial transfer without damaging normal neural function?
That's like targeting the nerves. I mean, beyond the philosophical question, there is this clinical therapeutical application, because do we target the neurons? Do we target the recipient cells? Obviously, targeting the neurons come with some level of toxicity that needs to be managed. But as Gustavo said, there is so many highly specific nature designed neurotoxins that could be used. And what we used in the paper was Botox as a general denervation mechanism that works very well. Other peoples have shown similar outcome, especially Gustavo with your clinical trial.
One of the things I did 15 years ago, I didn't propose Botox as a means of treating cancer, but today I'll be more aggressive. So what I did 15 years ago, I just did it because I wanted to know if the biology that I found in vitro and in rats and in mice existed in humans. And I did prove that. You put Botox into a prostate cancer and it starts killing cells. They undergo apoptosis. Let's take radiation therapy. So radiation therapy actually induces axonogenesis because it creates damage in the body. So radiation therapy, while it's killing cancer cells through DNA damage, at the same time, is increasing what could defend the cancer cells. So let's design a clinical trial, neoadjuvant Botox to radiation therapy, which would make radiation therapy more efficient, and we could even lower doses. That's a clinical trial in the waiting. Surface lesions in the cervix, in the bladder, in the skin, in the head and neck. Can you induce regression through denervation with Botox? I'm pretty sure it will. And these are things that can happen tomorrow.
And to speak about the generality, how broad is this concept for the audience? I mean, to clarify, I'm not aware of any cancer which is not innervated. Innervation in cancer is a general process. We are not talking about only few subtypes of cancers that could have nerves into their stroma. This is a general mechanism which applies to every cancer, from my knowledge at least.
Gustavo, you mentioned earlier that there's a need to move the discoveries into the clinic, and that very well aligns with one of the things that was seen as an outcome from our Cancer Neuroscience conference last year. But another thing that was discussed as part of a panel discussion at that conference was that there's a need to look at the way that clinical trials are structured. And there was like a consensus that there might be a need to have a slightly different setting for the clinical trials for them to best fit for this research field. What's your opinion on this?
I totally agree. We have so many tools we didn't have 3 years ago. Let's start using them. There has to be a faster way of doing things. Listen, we could use intermediate endpoints if necessary to look at things, or at least as guidance. It's dangerous sometimes to use intermediate endpoints. I'm a pathologist. I live with intermediate endpoints, but I fully agree. We can't start today and have the first results of basic things in 7 years. And again, because of what we have found, we could focus the clinical endpoints to very aggressive disease. Now let's define aggressive disease. Most biomarkers look at the cancer cells, and what they're measuring is the genetic load that this cancer has gone through. But if we also accept, I do biomarker discovery and I concentrate on non-cancer biomarkers. I measure the host. One of the things I measure is nerves. So if we design these studies, we first define what aggressive cancer is and how we're going to select these patients, and then concentrate on the very aggressive cancers, we'll have results much quicker. Because, prostate cancer is the leading cause of death for males. But do you know what the mortality for prostate cancer at 5 years is? 1%. Because we're culling a bunch of things that are not cancer and that have very little aggressive potential cancers. So I think that we need to focus on this population of very aggressive cancers and start testing neural modulation as soon as possible.
Thanks both of you for sharing some insights from your study with me. I'm glad that I had this opportunity to discuss such an interesting paper with the minds behind designing the study. Looking back, which would you say was the key experiment?
On my view, that's a mix of three elements. I think the Botox innervation in different type of setup, again, rats, mouse, humans, gave the big pictures of the story. The mitotracer brought the mechanistic answer, and the collaboration between Gustavo and me be an incredible key on the success of this study.
I'll say the key to this paper was the fact that Simon published another paper, and I sent him an email congratulating him. And he said, you need to come and give a talk. And we started talking, and he showed me the data that he had in breast cancer. And I told him, Simon, I have 50-year-old data I never published. I did all the generation experiments. I have all the transcriptomic data. And the key was that we connected, and in 2 years, we did all the data for this paper.
That's impressive. Well, Simon, Gustavo, thank you so much for taking the time to share your research results with me and with our listeners, and for giving us the opportunity to learn a bit more about the blooming field of cancer neuroscience.
Thank you for having us.
Thank you very much for the invite.
We really hope that you enjoyed listening to this fantastic research story and that you learned something new from us today. If you have any follow-up questions regarding this conversation, please don't hesitate to contact Simon or Gustavo by email. If you enjoyed this episode, you may be interested in the EACR conference on cancer metabolism to take place in Bilbao, Spain, between the 6th to the 8th of October 2026. You can find out more about the program on the conference website at www.eacr.org/conference/cancer metabolism 2026. Please note that the abstract submission deadline is on the 29th of June 2026. Please subscribe to our podcast so you won't miss an episode. You can find The Cancer Researcher on your podcast platform of choice. Subscribe and tell your friends about it. And don't forget that you can also find us on Blue Sky and Instagram at HelloEACR, and you can search for us on Facebook and LinkedIn. We welcome feedback and suggestions for other topics or preferred guests through our feedback form, which you can find on our website at www.eacr.org/podcast. Until next time.